Tonavir's kind of been known over the years as one of the more famous cases with Abbott. They had a form that appeared after they had been on the market for a year or two with their semi-solid capsules, and all of a sudden a new form started to appear in their product. So a new crystal form was found in the formulated product. A team of scientists went to investigate if any significant changes had been made in their manufacturing process. Until this time, no detectable quantities of Form 2 had been detected in the bulk drug lots. Soon after visiting the manufacturing facility, Form 2 started showing up in their bulk drug production facilities as well. So they moved the seeds from the product, and then they seeded their process, and then all of a sudden they had trouble controlling their final process. They were off the market, I believe. This was first in class, I believe, of one of the protease inhibitors, and obviously there was a lot of it was growing in sales very rapidly, and all of a sudden they had to withdraw from the market while they reformulated. They theorized that the compound adopted an energetically disfavorable conformation in the stable crystal structure, and that's why it didn't nucleate very well. To be honest, the data wasn't really good behind this. I mean, it was kind of a little hand waving, which is kind of what you find with a lot of times with these polymorphic forms. Didn't know, I mean, they theorized, but there wasn't an absolute answer as to what was going on. But they theorized that a heterogeneous nucleation occurred by a related substance that had that unfavorable conformation, nucleated it. This was by Bauer, who was in a different area, but at the same time, though, the guys in process chemistry published a paper, and they said the origin of Form 2 remains debatable. The fact was that this issue had to be addressed as soon as possible. And so there was a lot of urgency, and I knew enough of the guys from around there to know that, yeah, they were working a lot of hours. And what was interesting, though, is if you took it, it was a part of this study that Sarah June had done, and it was at one of the ones that's at extreme of differences in solubility ratio, and it was, if you calculated, it was about 0.8 kcals per mole. Theoretically, with polymorphs, they say that polymorphs will fall within 1 kcal per mole of one another, but the reality is most of them fall within 0.33 kcal per mole. So that brings me to my final system that is somewhat extraordinary, and so I called it the missing polymorph of melatonin agonist. But basically, if you remember Pasteur's experiments where he kind of led to some of our increased understanding of chirality and using tartaric acid, where he noticed under the microscope that there were half of the crystallites were of mirror image to the other half, non-superimposable, and it helped him infer, basically that they had, chirality associated with the chemical entity of tartaric acid. But in theory, if you took D and L tartaric acid and crystallized them separately, you could get the same forms out of each enantiomer. It's a mirror image, so they'd be mirror image crystals, but theoretically you could get the exact same packing out of them because there's the same molecules, they have the same conformational freedom and flexibility, they should produce the same forms. But in this case, the active enantiomer was produced by Discovery Chemistry. They decided that they would use this was in the mid to early 90s, and they went with the active enantiomer and provided for, and I've had this happen more than once, where to conserve the active molecule, we know biological systems are very sensitive to chirality. Crystallization, usually you do an achiral or non-chiral media, and so theoretically you should be able to learn what you need to from the inactive enantiomer. And so they in the study of the inactive enantiomer, while they were off doing their tox studies and the like, 3 out of 4 of the initial batches that were provided were metastable form 1. The 4th batch resulted in this much more dramatically stable form 2. It was extremely difficult to produce this metastable form after the discovery of the stable form 2. And in fact, I only was able to go back and produce the metastable form 1 time. Not surprisingly, that it came out of that it was from the oil, where I was actually able to get it to nucleate out as form 1. But it was much more difficult to get form 1 once the more stable form was produced. And that's not all that uncommon. I've seen it a few different times where you'd produce one form and then a next form and then a next form, and then ultimately you couldn't go back to the prior forms. It's been, Joel Bernstein again referred to that as being disappearing polymorphs, but it's been noted over history that this is not uncommon. There's a number of good papers where this has occurred. And some people say with ranitidine hydrochloride, which had a big patent dispute over it, but that was the case with ranitidine hydrochloride. So I had a disappearing polymorph in that case with the inactive enantiomer. But when I got the active enantiomer to work with, we never could reproduce the stable polymorphic form 2 that we knew existed from the inactive enantiomer. Even though we did thousands of recrystallization experiments and more than 10 multi-kilogram scale lots, each one of those produced the metastable form 1, which was quite surprising. So in that case, I called that the missing polymorph because we knew it had to exist, but we couldn't produce it. What happens sometimes is you run into a big problem, and sometimes attrition takes care of the problem for you. And that molecule, we stopped developing it. It was in phase 2 clinical trials, I believe. We stopped developing it, but it took, we actually out-licensed it, and then finally the company that was actually working with it gave us, after the patent had expired, I got approval from on the chemical entity on legal to go ahead, and actually the company that had purchased it from us went out of business, and then legal finally said I could publish on it. And so I had to wait 14 years, but ultimately if you wait long enough, you can publish it.
Question on this. So when you have enantiomers like this, normally the racemates have a much lower energy, and the racemate's the first one to crystallize out.
But these were purified. So they actually, the very final step was a chiral producing step. Yes.
But if what you had observed there is correct, then it should be self-resolving. No?
Well, that's interesting. And I'm not going to actually be able to go into that in detail. But yeah, you would think that, like Pasteur's experiment, that it would be a conglomerate. But it
should be so if you put the racemate in, then it should self-resolve, because one has a much higher or a much lower.
We won't have time enough to go into that here. But there's some, if you have interest in this kind of thing, read the paper. I went into a lot, probably more detail than I should have, but it's an interesting, really interesting system. But so after 14 years, I thought, my mind, that really wasn't that way, was it? And, my mind was just, there's no way in the world it would still behave that way. But it was one of those that just stuck in my mind. And so once I got the approval, the first thing I did was actually, I got out of research stores, I got like 30 some lots of the active enantiomer and looked at them all, and they had all stayed stable, and they had stayed in that metastable crystalline form for 14 years, and I was pretty surprised with that. And then I looked at so then I took the 2 primary lots, one of the active enantiomer and one of the inactive enantiomer, and then I just recrystallized them against, I believe it was like 23 different solvents as separate experiments. And surprisingly enough, the active enantiomer melted or formed the metastable form, the one that melted 127.9 degrees C each time. And the inactive enantiomer always crystallized out at the as a higher melting point in crystalline form. I didn't expect this to happen. I thought it was, I was amazed that that would occur.
So is these results published or?
Yeah, I did. Yeah, I'll reference it in here, but.
What I learned is two racemic enantiomer here. The physical property should be the same.
They should. You're absolutely right.
What I learned
Yeah.
it is not.
You would think they would be, but they weren't. And so I studied it, and I'm not going to go into all the details, but but part of it was, obviously, related substances being most similar to the drug itself are the most likely culprit. And but in this case, the purity was almost identical. They're both, one's 99.3% pure, the other's 99.5%. They had the same related substance profile, just slight, very small differences in the rel subs. Nucleation is what I'm going to say is very interesting, and we still don't fully understand it. I was able to take the two individual forms and make a racemate, but I did a lot of other studies that are published here trying to understand it.
It crystallizes the racemate.
It does crystallize as a racemate. Yes, it does. Yeah. Yeah, and so that actually makes some pretty nice cover art. So this is if you actually do the experimental, I should have left the experimental points on there probably, but it doesn't look as pretty if you do. But I measured the actual thermal phase diagram. But so in the one case, because it's the lower melting point, when I did the melt eutectic, it had a different eutectic because it was a different polymorph mixed in with the racemic. And when I did but your lower melting point form has a polymorph that's obviously missing because we know that it should form that polymorph. But in the other hand, we had with the inactive, once you formed the stable polymorph, the one that melted so much higher, you couldn't form other than the one time where I did it from the melt, or from viscous liquid, supersaturated liquid, was able to get it. But essentially you had a disappeared polymorph. It had become much more difficult to form that metastable form. And so I did publish it, a couple of, there's some, but this is the primary paper, but there's a lot of detail in there because I'd hope somebody would go back and try to reproduce the work for themselves and actually find that missing polymorph. I used a variety of techniques, but I did want to relate again through solubility and through calorimetry, the phase diagrams, and also the solubility analysis. To get to the main point, basically, is that it was almost like ritonavir also, where that metastable form was fourfold times more soluble than the stable polymorph. So it was pretty exceptional. So these ones that are really hard to nucleate oftentimes are the ones that have the highest difference in energy. The ones that nucleate the most, I'll get into that in a second, the ones that nucleate more readily, or the first appearing ones, are the ones that are metastable, and that's been known for years. But basically, melatonin and ritonavir, the late appearing polymorphs, or the difficult appearing polymorphs, if you will, were different by 0.8 kcals per mole. So they are at the extreme for polymorphs. Roy was 0.14 kcal per mole. That's the concomitant one where we had difficult controlling which form would grow. So late appearing polymorphs are nearly 4 standard deviations away from the average energy difference of the average organic polymorphic systems. And so I did do these energy diagrams using the combination of thermal analysis and solubility analysis across a wide variety of energies or of temperatures. But I'm just going to boil it down to, when it comes to polymorphism, it's all about nucleation and getting different conditions and getting forms to nucleate.